We are very fortunate this evening to have Dr. Jennifer Adair present her work. Please join me in welcoming her. So thank you for trudging in out of the rain to be here tonight. I have a tendency to wander, so I just want to make sure if I step away from the microphone, you guys in the back can still hear me. We're all good? Fantastic. So I wanted to start by giving sort of a background on what gene therapy is. And for those in the room who are a little more scientifically savvy, I apologize for the basic level of some of the introduction, but it's important to make sure that, for me at least, that everyone in the room can kind of follow what we're talking about here. So I'll introduce gene therapy, what it is, how we do it. Then I'm going to talk to you about the first patient treated with blood stem cell gene therapy here in Seattle and that clinical trial and its results. And then I'm going to talk about the lessons that we've learned using blood stem cell gene therapy here in Seattle and how that's getting us to a place where we can make this particular treatment approach, DNA-based gene therapy, available for patients worldwide. So I first wanted to start with definition of a gene. Many of you know you've inherited genes from your parents, and this is what defines who you are. Every cell in your body has a complete copy of all of your instructions. That's your genome to make you. Each individual instruction is a gene, and each gene tells a cell how to make one molecule called a protein. Now, even though every cell in your body has a complete copy of these instructions, you obviously have different types of cells, skin cells, liver cells, eye cells, that are doing something different. And the reason they are is because the instructions that they're using in that complete manual are different from the instructions your liver cells or your eye cells are using. So to illustrate this, I wanted to briefly show you this cell. There are a couple of different compartments in the cell. So in the middle, that orange circle we call the nucleus. That's where all the instructions are kept, the genome, the DNA. And I've illustrated this as a single line, and the pink piece is a single instruction, a single gene. Now the cell has sort of another compartment called the cytoplasm. That's where a lot of the function of the cell happens, not all of it, but a good proportion of it. So within your cells, you have these little proteins that will read the DNA instruction and turn it into a message, and we call that message RNA. That molecule then gets transported into the cytoplasm, where other proteins, translating molecules, translate the message into that functional protein unit that the cell needs to perform whatever duty it needs to perform. And this is a process we're going to revisit at least 2 more times in this talk. So when we add the word therapy, it's a treatment that's intended to relieve or heal a disorder. So you put therapy and gene together, and what you get is that we're therapeutically delivering a piece of DNA as a drug into patient cells to treat a particular disease. So there are a couple of different flavors of how this could work. In the first, we call it gene replacement. So there are things called genetic diseases. This is where one or more instructions is not doing what it's supposed to do. Either the molecule is faulty or the instruction itself is faulty, and so the cell can't perform the way that it needs to. Gene therapy in this context would actually replace the faulty version of the instruction with a correct version of the instruction to restore that function to the cell. Second flavor is called gene modification. So here you have an instruction and you have a function, but we want that function to work differently or operate at a different level. And here we would modify the existing instruction that's producing a molecule that's functional to change that molecule's function. The third flavor is called gene knockout. So in your cells, at times, certain instructions no longer need to be read. And in cases like cancer, oftentimes certain instructions get turned back on when they should be turned off. And gene therapy can also be used to turn off that instruction again to make sure that it's not working when it shouldn't be. And lastly, there's something called gene addition. So this is not replacing what should have been there or modifying what is there or taking away something that's there that you don't want there. This is adding something that was never part of the original instruction manual. And this can be to give a cell a new function that hasn't been described before. And one example of this is gene therapy to treat HIV, where we provide molecules that prevent cells from getting infected with HIV that normally wouldn't be in our genetic code. So as you walked in down the hallway, you passed 3 Nobel prizes. And I hope if you didn't take the time to look at them or haven't seen them before that you stop and take a look on your way out. And the first of those Nobel Prizes went to E. Donald Thomas, who's known as the father of bone marrow transplantation. So naturally in Seattle, we like to think about blood as a potential target for therapeutic effects. And in particular, my lab looks at blood stem cells as a means of delivering gene therapy. So what are blood stem cells? Well, if we took a drop of bone marrow out of any patient, we would see a whole bunch of different cell types. There's red blood cells, there's white blood cells, there's platelets, and all of these cells come from one cell in your bone marrow called the stem cell, or the blood stem cell, which is shown at the top of this tree. This is why bone marrow transplantation works, because you can destroy a cancerous blood in one patient and replace it with blood stem cells from another patient. It also gives us an opportunity to think about ways to deliver therapy through the blood system. So who here has donated blood before? Raise your hand. And just shout out the answer. How long do they make you wait before you're allowed to give blood again? 56 days. Why do they make you wait that long, you think? Exactly. So that's about the length of time it takes when you lose that much blood volume for the blood stem cells in your marrow to replace what you've just given up. And so they want to get you back to your threshold level that you were before you're allowed to donate again. And this is part of a natural process that your bone marrow stem cells, your blood stem cells, are creating about 1 billion new cells every day of your life for your entire existence. So in gene therapy, and specifically blood cell gene therapy that my lab pursues, we're really interested in putting our therapeutic genes, our pieces of DNA, into the stem cell at the top. When that cell goes to the bone marrow and then divides, every blood cell it makes is going to get a copy of that therapeutic gene and that therapeutic molecule. Now, a few weeks, or I guess months ago, Stan Riddell talked about T-cell immunotherapy here at the Hutch. It's using a very similar process delivering DNA, but in this case, it's delivering it to these T-cell lymphocytes that are shown here. So same strategy, slightly different cell population that we're targeting, but both blood cell populations. Now, I'm going to back up. Sorry, I skipped ahead too fast. Too much coffee today. We were thinking about, okay, how do we get these therapeutic genes that we want to transfer into the stem cells permanently? Because we don't want them to be lost as these cells divide. And the answer actually came from nature, and it came from a not so wonderful part of nature called retroviruses, and specifically retroviruses like HIV. So these viral particles, and I'm just showing you what the viral particle as a cartoon looks like up on the right-hand side, and they're not capable of making more of themselves on their own. If they exist as a single viral particle in nature, that's all they are, is a single viral particle in nature. The only way they can make more of themselves is to infect a cell, and usually the cell of a living thing, either whether it be plant, animal, in this case, human T cells. Once they infect a human T cell, then they will utilize that cell's machinery to make more of themselves. And to show you how this works, we're going to go back to our cell diagram. So in this case, we're looking at an HIV viral particle that's existing in nature that now is circulating in the blood of someone who's just become exposed to the HIV virus. That viral particle is going to attach to a protein molecule on the surface of a T cell, in this particular case for HIV, that will permanently adhere it to the cell, and that tells the cell, oh, I need to bring that virus particle in. That virus particle then gets internalized in the cell where it loses its outer coat, and the molecule that's inside, which is actually an RNA molecule, a message molecule that I talked about before, gets turned back into DNA by the machinery that is included in the virus. That piece of DNA the cell recognizes should be part of the genome, and so it permanently inserts it into the genetic material of the patient's cell. Now, that message that was in the virus has access to all the same molecules that our DNA has access to make functional molecules. So the same DNA reading molecules and proteins will read that viral RNA and turn it into messages. Those messages get transported to the cytoplasm, where the same molecules that our cells would normally use to read them and make proteins will read them and unfortunately make viral proteins. Those virus proteins then will package into new virus particles, and eventually the cell will get so overwhelmed by this process that it will die, and all of those new virus particles will be released to infect new cells. So antiretroviral therapies target this pathway to prevent viruses from doing this. But from a gene therapy perspective, we said, well, if we could hijack this process to, instead of delivering viral proteins, deliver therapeutic molecules, we'd have a way to permanently deliver therapeutic genes into patient cells. So what we actually did was take these viral molecules in a lab and we re-engineered them. So instead of having all these viral proteins, we took those out and replaced them with our therapeutic molecules, therapeutic pieces of DNA that are in a message form, in an RNA form. We wanted these to be able to get into a cell one time, and a specific cell, a blood stem cell. So we don't use what HIV particles look like in nature. Instead, we engineered the virus particle so that it can only infect one cell one time, and it specifically likes to infect blood stem cells. We then said, now we can expose blood stem cells to this engineered virus in a dish. It will get read by the molecules in the cell. It will get turned into messages that get moved to the cytoplasm, and then it will be getting translated and turned into therapeutic proteins. And it won't make new virus particles, so no new cells can be infected, but every cell that that cell divides into will also have a copy of that therapeutic genetic information as part of it. So at this point, I wanted to ask, who here thinks that this sounds super Mystery Science Theater 3000? We're taking HIV, we're getting rid of all the HIV particles, we're putting in therapeutic genes, right? Well, we've done it. We've done it in about 2,000 plus patients worldwide, specifically in blood stem cells. More patients if you count gene therapy to target other cells. And in all of these, we are trying to be very mindful that we want to specifically get these genes into one cell type. So the way that we're currently doing it, which I'm highlighting on this slide, is in a method we call ex vivo, meaning the cells that we're going to transfer these genes to, we take them out of the patient's body first, so that we're not giving virus particles to patients and hoping they get to the right cells. So for blood stem cell gene therapy, the way that this works is we need to get the blood stem cells out of the patient first, and we do that by one of a couple different methods. So we may go straight to their bone marrow, pull that bone marrow out. There's also in certain patients a way for us to not have to go into the bone marrow. We can actually give them an injection of growth factors under the skin once a day or twice a day for about 5 days, and that tells the blood stem cells in the marrow to come out of the bone marrow and to move into the peripheral blood circulation. And then we can draw blood from the patient's vein rather than having to go into their bone marrow in a procedure room or an OR. Once we have that blood or that bone marrow illustrated in the red drop there, we want to get the stem cells separate from all those other blood cell types that are there. Because our virus particle, in order for it to have a good chance of getting into a stem cell, we need to have a high ratio of viral particles to blood stem cells present. And if we keep all the other blood cells there, which are actually the vast majority of the blood cells in that droplet, the virus will be spending all of its time looking at all those different cells trying to find the stem cell. So we actually separate out the stem cells. And the way we do this is using a receptor that's specific to the stem cell molecules. So this is something that's on the surface of the stem cells that distinguishes them from all the other blood cell types that are present. And it, again, sounds a little Mystery Science Theater 3000. And the person who actually does a lot of this here, Fred Hutch, is sitting a few rows up, so he's kind of shaking his head and laughing at me. But we actually take a molecule called an antibody that knows what that surface molecule looks like. And we couple that antibody to a little metal bead. We then incubate the blood or the bone marrow with these beads, and the stem cells will attach the antibody and the bead because they express the marker we're interested in. And then we hold up a big magnet, and it pulls everything attached to the metal beads to the magnet and lets everything else flow away. And then when we feel confident that we've washed away all the things that are not stem cells, we take away the magnet, and what we've got is a pool of stem cells that we can then use to genetically modify for patients. And in all of these cases I'm talking about, we actually do this with the patient's own cells. So this is not like a donor to recipient case in bone marrow transplant. This is the patient's own cells that we're doing this with, so there's less chance of rejection or any complications associated with them recognizing the cells as foreign. Now, in All of these cases, we then take those stem cells in a culture dish and we expose them to the viral particle that we engineered to allow them to take it up and incorporate it into their DNA, into their chromosomes. And the goal is that we're trying to get as many cells to incorporate one copy of the therapeutic gene as possible. And this is permanently inherited in all of those cells. We can actually track using bioinformatic tools the exact location in the genome where these things will integrate permanently. And we can use that to do lots of studies into blood cell biology to learn things about what happens to these cells when they go into patients and how many of them will actually produce blood cells, if some of them will produce only red blood cells or only white blood cells. And so while we're using this as a therapeutic approach, the downstream benefit is that we're learning a lot about blood cell biology and development that we didn't know before because we're using this method that permanently modifies the genetic information. Once we have that done in a dish, we then need to give those cells back to the patient, and we do this typically through an IV infusion. However, as we mentioned, 56 days after you give blood, your bone marrow stem cells recover that blood loss. And so if you add more stem cells on top of that, they're going to get in your body and say, well, you don't really need us right now, so never mind. So we have to create a little bit of space so that your body thinks it needs stem cells and it needs those stem cells to produce new blood cells. And the way we do that is with something called conditioning. So this usually involves either radiation or a little bit of chemotherapy. It just is enough to create space in the bone marrow so that when we infuse these gene-modified stem cells, your body will accept them, they'll go into the bone marrow, and they'll start producing new blood cells. If this works, gene-modified blood stem cells will go to the marrow. They'll produce all the different blood cell types I showed you in that tree, and they'll do it for the lifetime of the patient so that therapy is there forever, and hopefully without any complications. Now, I mentioned that we've done this in several thousand patients now worldwide. Specifically for blood stem cell gene therapy, this is a list of diseases that have been successfully treated with this approach. And all of them are using different genes. A lot of them are gene replacement. Some of them are gene modification. What you may or may not notice or know about these particular diseases, I would say, how many of you in the room recognize a disease on this list? That's a good number. How many of you recognize several diseases on this list? It's a much smaller number of people. So when we talk about this, you guys aren't the only ones who sort of laughed when I said Mystery Science Theater 3000. We first took this to the FDA, or the European equivalent, the EUA. They kind of said, we're going to tell you that you need to test this in patients who really have no other treatment options. And so we were limited to relatively rare diseases where patients don't have a lot of options when they come into clinic or when they have their particular diagnosis. And then once we get through treating multiple diseases of that type with this therapy, they're then allowed to expand it to diseases that have more broader patient scopes and applications. And so we've now gotten at the bottom of this list to some cancers, which is good because there are lots of cancer patients. And what we've learned from these diseases is now being branched out to treat other diseases like HIV infection and sickle cell disease. And if you know anything about these particular patient populations, there are millions of patients affected by these diseases worldwide. Now, one other point that I wanted to convey is that it is not beyond us to recognize that the majority of those patients are also not in the U.S. And so we have to be mindful that these things that we're doing are going to be eventually translatable to the places where the majority of these patients exist. And that's something I'm going to revisit at the back end of this talk. Now, I wanted to briefly review. There are about 6 gene therapy, blood stem cell gene therapy trials currently happening right here at Fred Hutch in conjunction with the University of Washington, the Seattle Cancer Care Alliance, and Seattle Children's Research Institute. The first is to treat cancer, brain tumor in particular, called Glioblastoma multiforme, and this particular flavor of gene therapy is actually a gene modification. The second is a genetic disease called Fanconi's anemia, and this is gene replacement. The third is actually 2 clinical trials used to treat a combination of infectious disease and cancer, specifically patients who have HIV infections who have lymphoma related to their HIV infection. And that's a combination of both genetic modification and addition of new genes. The fourth is another genetic disease that primarily affects children and is also known as bubble boy disease. This is X-linked severe combined immunodeficiency syndrome. The vast majority of these patients are boys who are born with the inability to make any immune system whatsoever. Hence, back in the 70s, the best way to keep them alive was to put them in an immuno-restricted bubble. Here we are replacing the defective gene. It's one gene that causes this deficiency with a working copy that allows them to generate an immune system. And in the fifth trial, which is not yet started, but we're getting ready, we're treating another genetic disease called Wiskott-Aldrich syndrome, which also has some immune complications associated and primarily affects pediatric patients. But I wanted to really take you through a gene therapy trial experience, and to do that, I'm going to focus on the very first patient that I had the pleasure of working with and treating with gene therapy here in Seattle. And this was a patient who enrolled in our cancer glioblastoma multiforme gene therapy trial, and his name is Charlie. So Charlie was, at the time, a 56-year-old psychiatrist living in Alaska. He's shown here with his wife, Elaine. He also had a daughter, Shelby, who had just graduated from high school and was starting to apply for college. It was the summer in between, and they were enjoying a family picnic at a cabin on a lake in Alaska where they lived when something kind of odd happened. And it doesn't seem like it's that big of a deal, but Charlie had sort of been heading out to take out the boat, and he stopped for a second because he would normally always carry water with him. And then he started thinking about it, and he wasn't a coffee drinker, but he'd somehow managed to drink about 4 quad-shot espressos that day. And he wasn't feeling any different, and he also wasn't feeling very dehydrated or like he had to use the restroom or anything, and this struck him as odd. This is not typically his behavior. So he went back up to the house to share this with his wife, and by the time he got from the dock to the house, he was slurring his speech. He couldn't really make eye contact with her or focus on what he was doing. She right away said, "I think that we should go to the ER." And as is standard practice in the ER, when a person presents with these symptoms, they're given a CT scan of the head, and this is what Charlie's CT scan showed initially. Now, this is looking from up through Charlie's chin, so this is actually the left side of Charlie's brain, and this big bright spot is not supposed to be there. Now, just because it's a big bright spot, that doesn't tell anyone looking at it that it's in fact a tumor. The only way to really know that is to either stick a needle in and take a piece out, or to actually do a surgery to remove this particular highlighted region and then look at it under a microscope to know. Charlie wanted to do this at a place that had the expertise to do these kinds of brain surgeries, and so he came to the University of Washington's Neurological Surgery Center, where he went to surgery. They took out the mass and identified that he did in fact have a stage 4 brain tumor, which happens to be the most common brain tumor in adults, also known as glioblastoma multiforme. Now, this was in July of 2009. 1 month later, as was widely publicized, the same type of brain tumor killed Ted Kennedy. And so Charlie was seeing all of this and understanding that the situation was not exactly great. He did want to pursue therapy, and for patients who are diagnosed with this particular brain tumor, if they decide not to undergo therapy or any treatment, they have an expected survival of about 3 months. If they do undergo treatment, it's not trivial. So in addition to the brain surgery that they've either already had or have to have if they've only had a needle stuck in to take a piece out, they also undergo chemotherapy and radiation to their brain at the same time, followed by more chemotherapy. This is the standard treatment for anyone diagnosed with this disease, and despite all of that treatment, less than half of patients survive 1 year from the date they're diagnosed. Very few patients will live 2 years. This was obviously devastating news for Charlie and his family, and unfortunately, it wasn't the worst of it. About 50% of patients who have this brain tumor will turn on a gene, turn on an instruction in their brain tumor cells that should not normally be on, and that gene happens to undo all the toxicity of chemotherapy. To illustrate this, I'm showing you a computer model that we generated of Charlie's original brain tumor. And once we identified that his brain tumor cells express this protein, going a little faster, we knew that his tumor would grow pretty rapidly, and we anticipated we would cut the video off here when we expected that Charlie would no longer be able to survive the brain tumor mass, and we expected, based on this time lapse of growth, that that would happen in about 316 days from the date of his original trip to the ER. So the chemotherapy molecule in this case can go into the patients and it can try to get in, but this protein that the brain tumor cells produce just keeps blocking it out. So even though they're getting the chemotherapy and the radiation, it's not really doing anything. Now, if you wanted to think about this, it's only one protein in their brain tumor cells that's causing this block in the chemotherapy treatment. So the first thing people thought of was, well, let's make another molecule that will basically turn off that protein. A small drug that's not going to affect other tissues that we can give with the chemotherapy in these patients that will basically allow them to have the benefit of the chemotherapy on the tumor cells. And that molecule is called 6-benzylguanine. It's just shown here in the orange box. The first trials of this were conducted in the early 1990s at Duke University by a collaborator of ours named Henry Friedman. So he administered the molecule and he also administered the chemotherapy to the patients. They were able to show that the chemotherapy then went into the brain tumor cells and it started to have its cytotoxic effect. It was starting to do some damage to the brain tumor cells. Unfortunately, the patient's blood cell counts started dropping dramatically, because it turns out that this particular molecule, when you give it with chemotherapy, it is going to do some serious damage to any cell that's rapidly dividing. And I told you earlier that your blood stem cells need to make about 1 billion new blood cells a day, so they were highly sensitive to this combination treatment. Patients enrolled in this trial could get maybe 1 or 2 cycles of this chemotherapy when they normally should be given somewhere between 6 and 10 before their blood cell counts crapped out so much that they had to come off the treatment. So they were never able to see whether or not 6 or 10 cycles of this combination would make a dent in these patients' brain tumors. The small molecule with the red X was taken off the shelf because the FDA said, we're never going to get past this blood toxicity issue. This was back, again, as I mentioned, in the early 1990s that this was tested. The data came out in the early 2000s. Between 2001 and 2008, another investigator here named Hans-Peter Kiem, who's shown in the middle of this picture, said, I think that we can modify the same gene in blood stem cells to make them resistant to the sensitization drug. And I think we could do that with gene therapy. The idea here would be that then all the blood cells would have a shield against this combination, but the brain tumor cells would still be sensitive. Now, unfortunately for a few patients, not treated here but elsewhere in the U.S., there had been a few pharmaceutical errors where they had accidentally been given these 2 drugs and in really high combinations. And we knew from those patients that really the toxicity to the blood was the only major issue. They didn't have toxicity to kidney or liver or other tissues that may be detrimental when we talk about giving strong chemotherapy drugs like this. But the gene therapy angle also gave us the opportunity to administer the sensitization drug with the chemo and suggested that we might not have to give a lot of chemotherapy to these patients. We may be able to give just a little bit with the sensitization drug and really put a double whammy on the tumor cells without harming the blood. So the way that we designed this, because it's in adult patients, we decided to do the growth factor injections and then the peripheral blood draw, which is called leukapheresis. So in this case, Charlie and the other 10 GBM patients who were enrolled in the study to date all underwent this procedure. We collected their leukapheresis and isolated the stem cells using the same magnet technology I told you about earlier. We exposed them to a virus that encoded our protective gene. MGMT is the abbreviation for the gene that we're modifying, and the modification we're putting into it is called P140K, hence MGMT P140K. We exposed the cells to viral particles that encoded this gene modification, and then we infused them back into the patients after we gave them a little bit of chemotherapy for their brain tumor, because we knew that that chemotherapy would be toxic to the blood. It would create a little bit of space, but it's also going to target their brain tumor, which we want to make sure we're also trying to do. What we were able to show in all 11 patients is that these protected blood cells, stem cells, would go into the marrow and they would produce protected blood cells, and that this allowed patients to receive both the sensitization drug and the chemotherapy, and in some cases, as for example Charlie, up to 9 cycles of that drug combination without any toxic effects to their blood. So what did this do for those 11 patients that we've treated so far? It definitely improved their lifespan. we found, as I mentioned earlier, that only less than half of patients treated even at our hospital, at the University of Washington or Seattle Cancer Care Alliance, who don't get gene therapy, who get standard treatment, less than half of them survive a year. But 100% of the 11 patients enrolled in our gene therapy trial were still alive 1 year later. Some of them even lived beyond 2 years. And in the case of Charlie, he actually survived 6.5 years from his brain tumor diagnosis without any evidence of a brain tumor, was able to see his daughter graduate from college, was able to retire from his job as a psychiatrist and go on, enjoying his life. Now, unfortunately, about a year and a half ago, Charlie called me and said, "Hey, Jen, and I'm not really feeling that great. I've just been kind of down. What do you think?" I talked to Dr. Keem, and we said, go get your blood drawn. Let's see what's going on. He hadn't received any other therapy after our treatment in those 9 cycles. We couldn't see gene-modified blood cells really in his circulation, but we noticed that his blood cell counts weren't exactly normal either. So we had Charlie come down, and we took a better look in his bone marrow. And it turns out that there was a fraction of gene-modified or protected cells and a fraction of cells that were not protected. And some of those unprotected cells survived that chemotherapy, all 9 cycles of it. And they did accrue DNA damage during that time. And so one of the non-gene-modified blood stem cells actually turned into what looked like it was going to be the start of leukemia. So this was not great. It's definitely not from our gene therapy, but it's from the chemotherapy combination. The difference is that Charlie no longer had a brain tumor, so he was a candidate for a bone marrow transplant. And he happened to have a matched donor, his sister, who could provide that bone marrow for him. He started to undergo the procedure for a bone marrow transplant, and in general, that involves more conditioning. In fact, conditioning that's going to wipe out all of your existing blood cell production, because we want to get rid of the cancer cells in the blood, and then replace it with a healthy person's, in this case, his sister's. What happened was Charlie underwent the conditioning. It wiped out all of his bone marrow. He came back to get his sister's bone marrow transplanted. And as he was undergoing his clinical evaluation for that bone marrow transplant, someone had, part of the normal workup is another brain scan. And unfortunately, what they found was 6 different bright spots in Charlie's brain. And at first they thought this could be leukemia that spread to his brain. We did a second surgery to take a look, and in fact it was his brain tumor returned. Now, what was interesting to us about this was that he had gone a very long time without any evidence of a brain tumor. And what immediately happened before his brain tumor came back was this conditioning therapy for his transplant that wiped out all of his blood cell production, including his immune system. That, to us, suggested that his immune cells were playing some role in keeping that tumor at bay. This is not something we've ever had the opportunity to learn before in brain tumor patients. And Charlie's participation in this trial and long survival are what allowed us to learn that. That gives us new hope for combining gene therapy that I've just told you about with the immunotherapy you may have heard about from Dr. Riddell's talk a few months ago, to really provide double whammy treatments for these tumors that can eradicate all of the tumor cells in their brain. In the meantime, we've gotten patients like Charlie 6.5 more years of survival, which is not negligible. So what are we learning, or what have we learned since 2009? Well, we've identified another subgroup. So you've got patients who get diagnosed with glioblastoma, half of whom have this protein turned on that makes them not responsive to chemotherapy. Within that half, there's another 30% that actually aren't even going to respond to gene therapy. And so for those patients, we need to really explore more advanced therapies because we don't yet have a treatment option for them up front. We also were able to really refine the gene transfer process in these cells in a way that makes it amenable to other clinics that have the same infrastructure and facilities that we have here at Fred Hutch. And we have, in fact, transferred this protocol to the Cleveland Clinic so that patients on the East Coast don't have to travel all the way to Seattle to receive the same types of gene therapy treatments. We've adapted this similar approach to all the other clinical trials that I showed you on that Clinical Trials in Seattle page, including HIV and those different genetic diseases with Scott-Aldrich syndrome, bubble boy disease, and Fanconi anemia. So what are we doing next in terms of gene therapy for glioblastoma multiforme? Well, we need to treat 2 more patients to complete the phase 1 study, and we expect that we'll have to follow the last patient treated for 2 years to tie up the data on 1 and 2 year survival and be able to take this to the phase 2 or phase 3 studies. We're now implementing more advanced patient screening so we can find that subgroup of patients that we know won't benefit from gene therapy and offer them other clinical trials or other alternatives. We are exploring the combination of gene therapy with immunotherapy with other groups here at the Hutch as well as outside of the Hutch to really try and do some damage to these very devastating brain tumors. We're expanding beyond the Cleveland Clinic to other clinical centers with our protocol so that more patients have access to this particular type of treatment in the U.S. And we're looking for corporate partners to fund the phase 2 and phase 3 studies. So at this point, there is no grant funding from the NIH or any other entity that has the amount of money needed to do a phase 2 or phase 3 study. We have to look to corporate partners to accomplish those things. And it's also important to know that in this particular trial, the NIH grant that we did get to fund the phase 1 expired after we treated the 8th patient. We still needed to get to 12. So it's actually donations from the community that support what we call bridge funds at the Fred Hutch, and we were actually able to receive one of those to treat and to pay for the treatment of the last 4 patients on the trial so that we have the opportunity to present the data to corporate partners for phase 2 and phase 3 studies. And lastly, we've now combined, so this gene modification, protection from chemotherapy for stem cells, is actually what we coupled with HIV gene therapy for patients with AIDS lymphoma. So we think that in these patients, it's very unlikely, although we don't know for absolute sure, that their blood stem cells are not infectable by HIV because they don't express the molecules on the cell surface that the HIV virus recognizes. So we think that we could take the stem cells, blood stem cells in HIV-positive patients, provide them with genes that not only resist the chemotherapy they would get for their lymphoma, but also with genes that prevent them or any other cells that come from them from being infectable by HIV. And in that case, when we give the patient their transplant for their lymphoma with their own cells, those cells will go into the bone marrow and graft, will expand and produce blood cells, including T cells that HIV likes to infect, but those T cells will now no longer be infectable by HIV. And what we're trying to do is see if this will actually allow patients to come off of antiretroviral therapy and provide a lifelong control or possibly cure of their HIV. And this highlights a significant barrier. So again, as I mentioned, when we talk about HIV, we're talking about millions of patients worldwide, and the vast majority of them do not live in the U.S., as I'll show you in just another slide. What I'm describing not only sounds Mystery Science Theater 3000, but it's highly technically complicated, and it requires a multi-million-dollar infrastructure to accomplish. We're fortunate to have that here at Fred Hutch in Seattle. There are very few clinical centers around the world that have that technology available to them. And to highlight that, I'm going to show you, this is in 2014, the prevalence of HIV-positive adults across the globe. The darker the blue, the more HIV patients are there. Now what I'm going to overlay are the clinical centers right now that are capable of blood stem cell gene therapy. And you can't come up with a bigger disparity in where the treatment is and where the patients are who need to receive it. And this is something that from the very first patient, from the day we gene modified Charlie's cells to treat his brain cancer, has been sort of stewing in the back of my mind because I am the person who's in the facility working with this virus in these patients' cells to try and get them gene modified and get them back into the patient. And I remember thinking after we performed this in Charlie, there is no way, even if we cure this guy, that this is easily going to be transportable to every patient in the world who needs this type of therapy. And to show you why that is, I want to talk to you about all the steps that are involved in this particular process. So the current state of the art, you have your patient who either goes bone marrow or apheresis. That product has to come out. I told you we have to prepare it. We have to add the metal beads with the antibody on them to get them ready for stem cell isolation. We use very sophisticated equipment. That's the big magnet that's going to pull out the stem cells. Once we have those stem cells, we then have to take them back into a clean facility because we're going to put them in a dish. That means they're going to be exposed to the air in the room. We're going to add virus to them and other things, media, to help them stay alive while we're getting the virus to transmit its genetic information, its genes, into the DNA of those cells. That requires a lot of manipulation, and all of those steps increase the risk that those cells are either not going to become able to engraft in the patient, or that they're going to be exposed to an infectious agent that you don't want to give to a person who has potentially a compromised immune system. Because of that, we then have to really scrutinize those cells before we're allowed to take them back to the clinic and give them back to the patient. And that requires a lot of testing before they finally go back to the hospital where they're infused in the patient. And this, as I mentioned, requires clean rooms. For stem cells, it currently takes about 2.5 days to do this entire process, from the time the patient's in the clinic getting their cells collected to the time they're in the clinic getting their cells reinfused. And it all has to be done under this umbrella of what we call GMP, Good Manufacturing Practices. This is sort of the FDA's certification that we're really carefully paying attention to every single thing we're doing with these cells to make sure that we're limiting the potential for them to not be capable of going back into a patient and doing their job. So if we think about this, we either need to figure out how to make million-dollar infrastructures and clean room facilities in all the clinics around the world, which is not feasible, or we need to think about simplifying this process into what I originally visualized as a black box, something that people who are already in the clinic working with these patients would be capable of using, that is encompassing all of these 6 steps. And we had tried actually over a couple of years to link together different pieces of technology to try to accomplish this, much unsuccessfully, I might add. And then you see that device, I told you this is the big magnet that separates out the stem cells. The company that manufactured that device actually came out with a 2nd generation, which they hoped would sort of automate steps 1, 2, and 3. And this is what that device looks like. We used this device to automate steps 1, 2, and 3 in some of the patients being treated here, and it was obvious to me right away that this device might actually be capable of doing steps 1 through 6 if we were willing to put in the time and a little bit of engineering effort to make it do so. So what we've done in the last 1.5 years is take our steps 1 through 6 that we do for clinical patients here, and then in an experimental setting, we put them through the paces on this device that we had to re-engineer, reprogram a little bit, to see if we could actually get through the whole process and get a stem cell product that was capable of being infused into a patient. What we liked about this device, as you see in the cartoon here, this machine that's going to do the leukapheresis, it's designed on some similar premises and similar technology so that nurses who know how to use this machine, which is available in 3rd world countries, could in theory also operate this machine. And what we visualized was that each of the pieces of gene therapy, now you need a different virus with a different therapeutic gene depending on the disease you're treating, but these would be available as pharmaceutical kits. So they're sitting in the freezer in the pharmacy. You come in, you get your diagnosis of your brain tumor or your HIV. Your doctor says, we have this stem cell gene therapy treatment. You either undergo your growth factor and your leukapheresis or you get your bone marrow drawn. The nurse gets to put it on this machine where it's established protocol. She just puts the right reagent from the right kit on, and what comes off are gene-modified stem cells in a bag that just go right onto the patient's IV line. Now, we're not there yet, just so you know. In fact, we have still a long way to go. But what we have been able to show is that this concept is possible. We have made gene-modified blood stem cells in this box, and we've done it on top of a bench in a standard lab, not a clean lab. It requires this device and a few other pieces of equipment that are readily available in clinical centers around the world. It can be done on a countertop or a relocatable table that can shuffle between potentially patients' rooms. And it results in gene-modified cells, at least blood stem cells at this stage, that are the same equivalent product to what we currently do in our multi-million-dollar infrastructure. We couldn't put these into patients, but we were able to put them into some animals to show that they, in fact, could do their job, and they, in fact, go in and they do a slightly better job than what our standard products are known to do in patients. So what are we doing next? Well, we're optimizing this. We still, as I said, have a long way to go between what we're able to show here and what we have in the kit in our clinic for patients worldwide. This is new stuff. So if you go to look, you'll see that I've given a few talks on this. The actual scientific publication of this data is still under review. So you're getting it before actually my scientific peers have an opportunity to see it. We're in the process of trying to relocate this technology specifically to clinical centers that don't have experience in gene therapy to show that it is, in fact, a validated process that will work in clinics such as those. And we're doing this in specific disease contexts like HIV, where we know there's going to be a large patient burden right away. We are also taking this technology and trying to expand it to T-cell immunotherapy, as I mentioned. In this particular case, T-cell immunotherapy that is targeted towards tumors uses a lot of the same technology, viruses delivering genes that are going to tell those T-cells to attack a tumor in a patient. It takes about 2 weeks to do that in a dish or in a clean room facility. We can get it done in about 7 days, and if you imagine that in a facility like the Fred Hutch, you can only treat as many patients as you have the facility space available and equipment available. In the same footprint, you could fit a lot of these machines. And the part that touches the patient's cells is disposable. So it's not as if we have to chuck the whole machine once a single patient is done. And that provides something called scalability, the ability to increase the number of patients that we can treat. Again, it's not entirely ready to rock and roll. One of the other critical pieces that we're going to have to address in the next few years is how to make enough virus particles to infect that many cells and transfer the genetic material. Right now, that's the next bottleneck that we've identified that's going to require additional work by myself and other scientists in the field. So with that, I'd like to finish and just highlight, we have a pretty small group. I'm relatively a new faculty member here at the Hutch, but I've actually been here since 2008 working in other capacities. So I'm just starting to build up my group. I also want to acknowledge Hans-Peter Kiem, who is the physician I showed you in the picture with Charlie and myself, who really pioneered the blood stem cell gene therapy approach here at Fred Hutch and gave me the wonderful opportunity to come and get involved in these patients and their treatment, and really supported me in the conceptualization of this in-a-box gene therapy that you saw at the end. A few people in his lab that supported all of these studies. Definitely the Seattle Cancer Care Alliance, University of Washington, Seattle Children's Hospital are our clinical partners in these gene therapy clinical trials. We have to work closely with all that clinic staff to know, okay, what does this look like when I have to go back into a patient? I can't just walk in with a bag to a child's room with, let's say, bubble boy disease, and sometimes the nurse will look at me and go, what are you here for? So we have to, work very closely to know what we've got to do when we've got to do it. And of course, our patients and our families. Some people look at Charlie's story and say, how sad that after 6.5 years he still passed away from his brain tumor. But without his participation, we wouldn't have learned the next step, that now we can manipulate his immune system and the immune system of other brain tumor patients to potentially control their own tumor, in addition to getting positive results from gene therapy treatments that give them 2 or 3 times as much survival. Now, I put this up here not to say, look at me, I'm really well funded, because the truth is I'm not. I put this up here to say, I finished my Ph.D. in 2005. In the last 11 years, NIH funding has not changed much. And if it has, it's changed for the worse, not the better. We have to get very creative to fund these studies, and to do that, we have to reach out to a lot of different places. And that takes a lot of time. I spend about 30 to 40% of my time writing grants to get money for these particular studies, and that's 30 to 40% of my time that I'm not doing the science that you just heard about today. So donations like the Bridge funds that supported the last few patients' enrollment in our clinical trial, that also support new studies. The gene therapy in a box study was entirely funded by Fred Hutch, and their belief in me that I could make something like this work and that it would be good for patients for the last 1.5 years. And all of those funds have to come from external sources as well. And with that, I want to thank you for staying awake this long. I'm sure most of you wanted to be out in the rain or in traffic. And I'm happy to answer any questions you might have. Yes?
I just have a whole stack of questions because
Great.
It's fascinating to hear, but I chose one based on...
Can we hand you a microphone just so everybody has the opportunity to hear?
I chose one question based on the sort of cost you're looking at to create the black box, because I realize that gene therapy has a lot of other benefits outside of this, and your observation that most of the HIV patients, for example, are in areas of the world that don't have a lot of money and aren't likely to be able to support the companies who would otherwise be covering some of those costs. But one of the things that we have in this country is a very substantial number of people with too much bad cholesterol. And and there is a small group of Italians apparently who have the ability to produce supergood cholesterol, described as liquid dreno for arteries. And this is actually expressed by their genes. So if you were able to take that gene, I think you'd find a significant number of people who'd be very happy to have supergood cholesterol inserted in their gene collections. I'd be an early volunteer on that.
What's your name? I'm going to write this down.
But it seems like it should be possible, and with that sort of exposure, you have potentially much greater funding than you would from people suffering from HIV in Southern Africa.
Right. And, there's lots of possibilities. Again, there's always this notion that you have to demonstrate the proof of concept that this will work. We have to show for all those flavors of gene therapy that it will work and it won't change the risk to benefit ratio of the treatment. And so to really demonstrate that initially, we have to do it in patient populations that don't have any other treatment options. There are many cholesterol lowering drugs and foods available on the market that we'd be competing with. And a lot of patients would say, take a pill or have my bone marrow harvested, my stem cells collected, viruses put into them, and then have them reinfused after I've gotten chemotherapy, which is not anything that would be standard treatment for cholesterol levels today, is a stretch. So definitely, and the other component of that is that the way that particular molecule works, it has to be secreted, so it has to also be in circulation. So now we're talking about a therapeutic gene that's not doing its job in a cell, in a confined compartment, but being released to be exposed to the rest of the person's body. And we would need to demonstrate that we're definitely not increasing the risk of the health of the patient by doing it that way. There are more places where we feel like we could implement something like gene therapy, then we actually have scientists who have the time and ability to write the grants to pursue. I wish it weren't the case. You should all become scientists and gene therapists, is what I'm trying to say right now. But certainly it's something, at some point we have to take the step and say, now you need something that, could potentially impact bigger health burdens, or for the corporate partners, have a larger market niche. Sure.
Thank you. This was a very great talk. I have a question, and I was wondering, why do you have to take out the cells, put them in your machine, apply this whole process? Why can't you just flood the patient with your virus and...
Do you want to be flooded with virus? Even engineered virus particles? Part of the answer is, that I can imagine myself trying to explain this to a patient in a conference, and I imagine that half of the patients that we've treated probably wouldn't have signed on if we'd have said that's what we were going to do. The second part of it is, I told you that we engineered the particle to prefer to infect stem cells, but that doesn't mean those are the only cells that it infects. And when you think about it, there are billions and billions and billions of cells in your body. The fraction of those that are blood stem cells is minute. If we injected virus into patients, even if we injected it into their bone marrow, the likelihood that more than just one or two or a handful of stem cells are going to get the gene modification is very low. So we're trying to hedge our bets and get as much bang for our buck as possible. So we improve safety because we limit the possibility that these viruses are going to transfer their genetic material to another cell. And most importantly, the FDA cares about sperm and egg cells. Okay? And the second piece of that is that we maximize the number of stem cells that get exposed to the virus as opposed to off-target cells. And that's to create therapeutic benefit. We can't say at this point that if we only got our gene therapy into one stem cell, that would be enough. We know that it wouldn't.
Great. Thank you very much.
Welcome.
I just wondered if you noticed any difference in treating different ethnicities with this.
Well, no, but we haven't really treated enough patients to be able to say that we have. In our brain tumor trial, the vast majority of our patients, because of our geographic location, are Caucasian. We have about a 60 to 40 male to female ratio. I think we have one person of Asian descent, one person of Hispanic descent. It's just not enough statistically valid data to say that we notice differences, at least from the gene therapy perspective. There are lots of other groups that have documented differences in blood stem cell type or ability to transplant donor blood stem cells into patients of different ethnicities, for example. It's something that will accumulate more data as we treat more patients for all of the different diseases that I put in that list. It's something that people are paying attention to. The NIH, in fact, requires all clinical trials to document the race and ethnicity of patients who enroll to make sure that we're giving a fair opportunity to every population that's available. But we just don't have the data yet to know if there are differences from a gene therapy perspective. Is the therapy inheritable? So the question was, is the therapy inheritable? Not when we do ex vivo blood stem cell gene therapy. But if the therapy were to happen to get into a sperm or an egg cell, then it would be inheritable. But the way we do it currently, we never give eggs or sperm cells the opportunity to see that virus because we're doing it outside of the patient's body with just the purified blood stem cells. If the blood stem cell were to produce a blood cell, and let's say that person got cut, and you happen to be there trying to stop their bleeding and you got a little bit of their blood, your immune system would recognize that those cells belong to someone else and it would eradicate them. Yes?
Thank you. During your talk, you made a distinction between gene therapy and immunotherapy. What is the difference between the two?
So there's a couple different flavors of immunotherapy, and I'm not an immunotherapist, so I want to throw that out there right now. I'm learning a lot. But in some cases, immunotherapy means taking immune cells from the patient who has cancer, who those immune cells already recognize the tumor as foreign, and basically bulking them up so that the patient gets a really large dose of those, what we call tumor infiltrating or tumor reactive T cells. The other type is similar to what we do with gene therapy. In fact, it is gene therapy where they purify the patient's T cells and then they insert a gene that makes those T cells recognize the tumor as a foreign antigen. There are a few more flavors that I don't know well enough to describe, but those are the two general flavors. So that's why I kind of grouped immunotherapy different from the blood stem cell gene therapy that we're talking about. In theory, sorry for those people who are still waiting to ask questions. In theory, we think that we can actually insert the same molecules that target the tumors to the blood stem cells and just have them only made in the T cells when the T cell develops from that blood stem cell. And then the patient will have immunotherapy that would last their lifetime against their tumor, but we haven't yet proved that concept. Yes?
On the regulatory side, I'm wondering with the benchtop, the regulations of the in developed countries and developing countries are different for acceptance, and that, from what I understand, WHO has different requirements. Mm-hmm. And I just wonder if you're looking at those, or is anyone looking at those, or the African Union requirements for acceptance, because that's different than GMP, from my understanding. Right. And then the second question I have is, what about allogeneic cells instead of the gene therapy? Are you looking at that where it'd be more like it wouldn't be personalized?
Okay. So there are two questions here, which I'll repeat. I also want to say that I'm going to get to the question from the person in the hat and you're next, so just be ready. So the first question was, obviously the regulations for cell therapeutics and other drugs, for that matter, are not the same between the European Union and the USA, let alone between these countries that are less developed or that have separate regulations. And the World Health Organization is who kind of oversees a lot of that umbrella, and you're absolutely right, there are big differences. So gene therapy in Europe right now at those centers has to be done under a different set of regulations than gene therapy here in the US. We have several large associations. There's a European and American Society for Gene and Cell Therapy now, which are very closely cooperating to try and make sure that we can bridge some of those gaps. So that we could actually take our therapies not to the other side of the U.S., but to the other side of the Atlantic or Pacific Ocean. There's also a Japanese Society of Gene and Cell Therapy, and we all work together with our governing regulatory bodies. I actually went and participated in a workshop that was sponsored by the American Society called Standardized Pathways in Gene Therapy to meet with the FDA to say, okay, you know, each investigator who proposes gene therapy to you gets a different set of reviewers, and so the criteria are different for each of these protocols. We want to align this so that we're all following the same standards, because until everybody in the U.S. is doing it the same way, we can't possibly bridge the gaps between ourselves in Europe or ourselves in Africa. It's something that we definitely have an eye on. I don't know that we have it perfectly right, and we haven't solved it yet, but I think it's somewhere that we're making positive progress towards. So then the second question was about allogeneic. So allogeneic means cells from someone else. Autologous means cells from yourself. So everything I talked about in my lecture today was in the autologous setting, so taking blood stem cells from the patient and returning them back to the same patient. In the allogeneic setting, probably the best described thing that we think about is something called umbilical cord blood. So cord blood is something that, you know, when you have a child, the placenta contains blood that is sort of shared between the mother and the infant, but it's actually pretty rich in blood stem cells on a per cell basis. And it's not something that requires someone to go through a bone marrow transplant or a growth factor injection to achieve, but it is not self, unless potentially you're giving it back to the baby it came from, but even then there's potential, you know, a few differences between the baby and the mother. So that is a possibility for patients who can't undergo stem cell harvest or who don't have good stem cell pools. There's definitely initiatives to look at whether or not we could provide gene therapy to cord blood stem cells and provide those to patients. The issue, one of the big issues there, is that while there's more stem cells on a per cell basis, there's very few cells because it's a very small volume. So it could probably be more easily incorporated into treatment for children who are smaller, but not necessarily for adults who need large numbers of stem cells to accomplish the therapy. So that's probably the best allogeneic source that we think about. From donor to recipient, it hasn't really been explored much, because when you start thinking about transplanting a donor cells into someone else, there are a whole different set of criteria that have to be followed. So even in the best circumstances, like I mentioned for Charlie that he has a sister who is a match. You can't get much better than that, to have a sibling who is a match to you. A lot of patients don't have that available. And the conditioning you go through there, you have sort of a competition. You have to not only create that space, but then you have to sort of put each set of blood cells' immune system in check. Because if the donor cells come in and their immune system recognizes you as foreign, then that graft that you got will attack you, and that's something called graft versus host disease. If your cells recognize the incoming stem cells as foreign, then you get something called graft rejection, meaning your immune system will wipe out the incoming graft. To avoid that, patients who undergo allogeneic transplants get what we call myeloablative conditioning, means enough conditioning to wipe out all of their existing blood stem cells, blood cell production, and immune system. That's not the kind of conditioning we're doing in gene therapy right now. Gene therapy patients, we give just a little bit of conditioning to make space for their own cells to go. So if we thought about doing this from a donor perspective in the allogeneic setting, we'd have to be thinking in the mindset of a full conditioning regimen, and that's something that we actually have an advantage for in gene therapy the way we're doing it currently, that we don't have to give that. You're welcome. So I have a question that's been waiting very patiently up here.
For any young scientists out there, do you have any advice for starting any good educational choices?
So I don't necessarily think I'm the best person to give early career scientific advice, because, and I'll tell you why. I thought for a while I was going to be an Air Force pilot, until I found out that as a woman I'm actually too tall to fit into their standard uniforms. That really made me mad. And so then I, did something silly. I was in high school. I thought I really like science. But I like to argue with my teachers too, and so my science teacher actually told me I should be an attorney. So I was looking at law school, and then I started dating this guy who happens to be my husband of 19 years now. And he was going to go study biology, and I thought, I'll just study biology with him. I really hadn't thought about it. Probably, some would say not the wisest choice at the time, but I don't think it turned out too bad. I really got fascinated by science because of him. I had no idea what a Ph.D. was or graduate school or anything like that. I came from a family of teachers and steel mill workers from the Rust Belt in the Midwest. And what I did was I said, okay, I don't want to manufacture windshield adhesive for GM or do any of the steel mill related chemistry stuff that my science degree would allow me to do in that area. So without really knowing what I was getting into, I took a job as a research technician at the University of Pittsburgh School of Medicine. And that was my first exposure to a scientific lab after college. I was just totally impressed. I thought, this is really cool. Like, these are the people who are making the discoveries that make the new drugs. But I didn't know how to get into graduate school or what to do with that. And so I just talked to people and decided that I should just go for a Ph.D. By that point, I was already married and had 2 kids, so it was a different, most entering graduate students are still single. It's a different set of criteria for me in thinking about which graduate school to go to. And so I ended up in Eastern Washington at Wazoo because Pullman, for what it's worth, for you Husky fans in the audience, is an affordable, excellent scientific program for a young graduate student with a family. I had a fantastic time there. Most Ph.D. programs take 6 years on average now, and I finished mine in 4.5. Had a wonderful time there. Came out of there and said, basic science is great, but I don't see how me spending hours and days and weeks at a bench working with cells in a dish is really going to make a difference in patients' lives. And so I did something stupid again. I decided after you do your PhD, you have to do a postdoctoral study. So I thought, well, if I want to treat patients, I'm going to have to get an MD too. So I moved to North Carolina for a postdoctoral study that really was in nothing that I was interested in scientifically. It was to establish residency so that I could try to go to medical school, one of the fine institutions available on the East Coast. About the time that I'd been accepted to 3 of those medical schools and was deciding which one to go to, my wonderful husband and my then 3 children at the time said, we don't think this is a smart idea, financially or time-wise. And so I kind of went, well, crap. How do I do what I want to do and make an impact in patients' lives with science without an MD? And I talked to a former mentor of mine who's used to be the head of the bone marrow transplant and pediatric program at St. Jude Children's Research Hospital, actually. And he said, oh, you don't need an MD. Now, science is primed. Like, we recognize there's all these great discoveries at the bench. There's all this clinical need, and we have no one that wants to put one foot in each corral. The MD PhD program, which is an actual thing people do that takes like a decade, was supposed to do that. But they were finding that MD, PhD people were either studying basic science as PhDs or full-time clinicians as MDs and not necessarily bridging that gap. And so it was just finding the right opportunity. I wish I could tell you I did all this homework and I identified the Fred Hutch as a place. Now, mind you, this is 2008, so I'm getting ready to come here to do gene therapy in Charlie. I wish I could have said I did research. I knew this was the home of bone marrow transplantation. I was super excited about the science. I wanted to do gene therapy. That's not what happened at all. I was totally lost. I had no clue what I was going to do. A friend of mine that I knew from Wazoo in Pullman, another graduate student, had come here to work with Hans-Peter Kiem. He knew he wanted to do gene therapy. He happens to be the godfather of my youngest child. He came to North Carolina to visit us. We went out to our local pub to celebrate his visit, and he said, well, what are you going to do? I said, I don't know. And he said, but what do you want to do? And I said, I think I want to do something more directly related to making a difference in patients' lives. I think I want to run a clinical trial. And he dropped his beer on the floor and said, holy crap, we just got this NIH grant to do this brain tumor trial, and we need someone who can understand the science, who's got an interest in learning the clinical side, that can basically get this from start to finish. And that was how I ended up here 8 years ago. The advice that I can give you from that is don't try to connect your career dots in the forward direction, because it will never work out that way. You'll only be able to stand where you are, look backwards and say, oh, that's what I did right that got me here, and I'm happy. it was being mindful of what I knew I wanted to do, being okay with the fact that I didn't fit in the standard box of a researcher or a clinician, and talking to people about that. It wasn't me finding that opportunity, it was me talking to people that then presented the right opportunity for me. So network, communicate, tell everyone, even people who have no idea what you're talking about, what you think you want to do, because you never know when someone with a different experience is going to present the right idea to you at the right time. I'm sorry, that was the longest answer so far. Yes.
So what's the order of magnitude of funding? I'm talking about money that you require to run through one of these trials.
So keeping in mind that right now the onus is on us to really demonstrate proof of concept, and we have to do a lot of extensive testing on blood from these patients after they receive treatment, on the cells that go into the patients, because we have to establish what the standard should be. So on average, depending on the patient and what they're being treated with, patients in the GBM trial, it costs somewhere between $90,000 and $140,000 per treatment per patient. For patients in the HIV study, it'll be a little bit more, probably closer to $150,000 to $175,000. Patients in our Fanconi anemia trial, it's about $35,000 to $40,000 because each patient population is also different. So multiply that times 10 to 12 to 15 patients needed for a phase 1 study, and then add on that I have to figure out how to pay myself while I'm doing this, which is where our salaries come from our grants that we collect. Then you get, a standard NIH grant has a cap of about $2.1 million. You're basically able to get there. The other foundations that I showed that were not part of the National Institutes of Health are all much lower dollar values. So there we're talking about not even a phase 1 study. We're talking about some bench experiments to cover the cost of the proof of concept experiment with the gene therapy in a box. I can tell you that I've probably spent about $120,000 in a year and a half generating the data for that paper for the proof of concept of gene therapy in a box. Phase 1 trial, we hope we can get away with an NIH grant to do that, but I don't know yet because we haven't gotten funding to do that.
So you're basically saying you need about $3 million to run one of these things?
A phase 1.
Later one's more expensive?
Yes, they are, because they require much more patients. That's why we have to look to corporate partners, because they will exceed the cap for what we can get in an NIH grant. Yes. Did I miss someone? No? Okay, go for it.
You've established the new treatment protocols. What has been the response of insurance companies to the idea of paying for this?
Yeah, so currently, we, in our phase 1 trials, for the most part, we can pay for all of the treatment associated because we're demonstrating. Now, the GBM trial happens to be a unique exception to that rule, because the chemotherapy those patients are getting is the same chemotherapy they would get as part of their standard treatment. What's different is the stem cells that are gene modified and the sensitization drug. Now, we were fortunate the sensitization drug was actually provided for free for this clinical trial by the what's called the Cancer Therapy Evaluation Program. So even though that drug was taken off the market and the company that produced it is no longer in business, the National Institute of Health and the Cancer Therapy Evaluation Program said, somebody might someday figure out a way to get past this, and so we're going to retain all of this pharmaceutical stock, and if you write us a proposal of how you think you can get around that, we'll dole it out to you for free. So that drug was actually provided for free. And then what we do is when the patient is diagnosed, we know that we have a molecular analysis that says, hey, their brain tumor is resistant to chemotherapy. We present that to the insurance companies, because for those patients, when they know that, they don't have an alternative treatment option available. So then we can say, they don't have an alternative treatment option. We're going to pay for all the new stuff that gets added on to this, but we want you to pay for their standard chemotherapy, their standard radiation. And so the 11 patients treated all had insurance cover that standard piece, and then we paid for the research components of that. I will say that we were not successful in securing insurance approval for all of the patients who wanted to participate in the study. So we actually had about 16 patients that wanted to participate. One patient wasn't able to go forward because of some complicating health issues that disqualified them for the study. And then the other 4 patients, the insurance said no. Yes. 2 more questions. Step right up.
So I'm just curious what kind of corporate partner you're going to find. I am a clinical research coordinator. I've been doing it for about 30 years, and drug companies always want to know what's in it for them.
Exactly.
And you're not, none of the therapies that you've described are tied to a particular drug.
So we actually have identified some. So right now, if you look at in the healthcare industry, new companies, you might actually be surprised to see that a good proportion of them are cell therapy companies and companies that include gene therapy approaches. So there are actually 4 companies that I could list off the top of my head that are not yet publicly traded, but have all initiated in the last few years that are targeting gene therapy of specific diseases with specific approaches. There are about 10 more in the works, and those are likely the corporate partners that we'll be joining with. That said, there are big companies like Novartis and a few others that you may be more familiar with who have already partnered with institutions to move some of these cell therapies and gene-modified cell therapies forward. Really, once you demonstrate that you have a good impact on human health, that's pretty much the key piece. After that, there are lots of opportunities. We talked about cholesterol, and all they need is to kind of get their foot on it soon, and then they can start thinking about expanding this out to diseases with much greater market niches. But not just in the U.S., also in Europe, there are a number of companies as well. And cell therapy is a growing field at the moment. One more.
Hopefully you're going to get a corporate partner.
We actually already have one who really likes the gene therapy in a box piece, and as well as the piece associated with our treatment of some of the genetic diseases. And we're talking to a few others about expanding that. It's not always official. I mean, there's lots to be done. There's always a balance, too, when you develop, when you have a corporate partner, that to some degree, then the research becomes very directed, and you sort of lose a little bit of that ability to say, wow, look what this did to the patient's blood stem cell development. And so we're always trying to balance making sure that we are taking the steps to get this to more patients worldwide and not losing the creative freedom that we have to explore new options that otherwise wouldn't get looked at because they don't funnel into that immediate market niche.